Chromosome jumping. It is a useful method to study certain DNA genes by jumping over unnecessary sequences. In the first step, the genomic DNA is partially cleaved using a restriction enzyme. This cleaving creates several fragments of DNA having different sizes. Now these fragments of DNA are circularized. The enzyme ligase stitches the ends of each fragment together. This creates circular DNAs. Remember, a DNA marker is added to each fragment during this circularization process. Now the circular DNAs are fully cleaved using another restriction enzyme. This step is crucial because it removes the bulk of DNA and leaves behind only those sequences that are near to the marker. The remaining pieces of DNA are then placed into suitable vectors. Remember, these remaining fragments represent the junctions of original DNA. This way, they grow in those vectors and form different colonies. Finally, a probe of known sequence is used to screen the colonies of opposite sequence. This helps to identify the clones that contain the target DNA sequence, which is located away from the starting point in the original DNA. So this method allows scientists to bypass large sections of chromosomes and jump to the target gene without cloning every intervening gene.